Okay, why don't we get started? Welcome to our science chats at the IGB. I'd like to thank you all for coming and thank our speaker today, Professor Amy Wagoner Johnson, who is a professor in the Department of Mechanical Sciences and Engineering. Amy also has an appointment at the IGB as a member of the Regenerative Biology and Tissue Engineering theme and is involved also in a new group that's exploring new ways to study women's reproductive health. Amy also has an appointment in the Carle, Illinois College of Medicine and was recently named the department head of the Department of Biomedical and Translational Sciences. Amy received her bachelor's degree from Ohio State and then her master's and PhD from Brown University. She's won numerous awards, including a Chair of Excellence in Nanosciences Foundation in Grenoble, France, and she has been appointed a Center for Advanced Studies Associate. She's known for her convergence research using mechanical sciences and innovative materials to address a number of issues in biomedicine related to bone, in the environment related to coral. And today she will be talking about the micromechanical and microstructural characterization of collagenous tissue for applications in preterm birth and for diseased and damaged tissue. Amy?
All right, thanks, Jean, and thanks everybody for joining us today. I've never given a Zoom seminar, so this is a first, but I'm excited for the opportunity. So I am going to go into some detail about what the heck my title means in a couple minutes, but first I want to just thank my collaborators who are listed on the page here. Professor Toussaint in Mechanical Science and Engineering and his former grad student who's now at UC Boulder. Also, Professor Toussaint has moved to Brown University where I did my PhD, so some nostalgia there. And then my graduate student, Amir, who did the nanoindentation and a lot of the image processing at the end of this. Also, I want to thank Heidi Phillips in Vet Med and Barbara McFarlane, who's a professor and nurse midwife at the University of Illinois, Chicago. So I'm going to talk a lot about this image that you see here today. You'll see that over and over as we go through the talk. And just to tell you what it is right now, this is non-pregnant rat cervix in cross-section imaged by an imaging modality called second harmonic generation imaging. So I'll go ahead and get started if my slides advance. Okay, so I want to say a little bit about what my lab does, and Jean mentioned this a little bit already, but I'll just go ahead and do my version. So my work focuses on biomaterials and biomechanics in general. So for those of you who might not know what a biomaterial is, so this is generally for a broad audience, it's any material that could be man-made or made by an organism. And the function is really to interact with any biological environment. So that could be inside the body, interacting with tissue or engineered tissue, medical implants, or it could be the environment, like materials for coral restoration. And then biomechanics is the study of structure and function of the mechanical aspects of biological systems. And that can span length scales from the whole organism down to the molecular level. And generally we use methods in mechanics and physics to do those studies. Excuse me. As Jean said, I've been part of the regenerative biology and tissue engineering theme really since it began many years ago. And in that regard, I've been working on tissue engineering for bone. And we do everything from manufacturing the scaffolds using 3D printing and improving manufacturing to materials processing, so more at the material scale rather than the structural scale. We've done in vivo studies and we analyze those studies using micro-CT for 3D imaging. And we've also done some mechanics of these materials and structure, so materials behavior. So stemming from this work, on this ceramic system for bone tissue engineering, we have a new project that is related to coral regeneration. And so there we're developing materials for coral regeneration and trying to understand how the coral larvae interact with the materials and surfaces they come in contact with before they become the coral animal that you see there. And then finally, what I'm going to focus on today is work in the area of soft tissue mechanics. And thus far, it's been mostly related to preterm birth. And we've done mechanical characterization, including indentation, which I'll talk about today. So this is a schematic of indentation. Also imaging the 3D structure using SHG. Again, I'll talk about that today. And then we've also done some finite element analysis. And this fits in with the new group that Jean mentioned, being led by Jody Flaws and Indrani Bacci in Vet Med, environmental impacts on reproductive health. So we hope to incorporate some of the techniques that I'm going to talk about today to understand how toxins in the environment affect reproductive tissue. Okay, so back to this long title. I realized after the fact that I should have just kept it much simpler, but I'm going to tell you about all the words in this title. So micro in this case, we're doing this at the micro scale, meaning small scale. We need a microscope to see what we're working on. Mechanical, meaning we're looking at how these tissues deform and what kinds of forces are required to deform the tissues. Structural, meaning what is inside the material that gives it the mechanical behavior that we observe. And characterization, so how do we describe the structure and the mechanical characteristics that we see. In this case, we're focusing on collagenous tissue. Really, most tissue has collagen in it. And in fact, we're focusing on collagen because the imaging modality that we use has a special property that allows you to see collagen really well without using stains that are often needed for other imaging modalities. We can use these tools to understand healthy, diseased, and damaged tissue, but today I'm going to focus on applications in preterm birth. Okay. So most of you, I'm sure everyone has heard of osteoporosis. Most of you probably know that osteoporosis is a disease that affects bone, and it affects elderly people. And what happens is the density of bone changes, and there are some compositional changes, and those changes result in weakening of the bone and can result in things like hip fracture in older people. And this can lead to a variety of complications that are can be life-threatening, actually. And so here, this is a prime example of how the tissue structure or microstructure influences mechanical properties and signals disease and damage in the tissue. Another example of this is with tumors. So a lot of times physicians will palpate tissue and they'll feel around for a lump, and they can feel that lump because it has different mechanical properties. Specifically, it has different stiffness. And it has a different stiffness because the internal structure of that tumor is different than the surrounding tissue. So that might mean it's more dense with cells or more dense with other fibrous tissue. But in any case, the structure has changed, and therefore it's changed the mechanical properties, and that's a marker of disease. Another example is in the coronary artery. So here I'm showing two images of two different layers in that tissue. And the graphs below show stress versus strain curves. And so for those who may not be familiar with that, that is a measure of the stiffness of the material. So the steeper the slope, the more stiff the material. So you can see on the right-hand side, this tissue looks like it's been disrupted. Maybe there's some damage, maybe it's less dense, and therefore the corresponding curve has a lower slope, meaning it's less stiff. So again, just another example of structure, the connection between structure and mechanical properties and disease and damage. So now I want to go directly to how all this relates to preterm birth. So infants, so 40 weeks is full term in pregnancy, and infants born at 37 weeks are twice as likely to die in their first year. So only 3 weeks early, they are more likely to die their first year. Premature infants, so less than 37 weeks, can have major developmental disabilities and health issues that can last their entire lifetime. So these range from things like asthma to things as severe as cerebral palsy, hearing loss, etc. And so this is a major problem with these babies, and it's a burden to both their families and the healthcare system. And in fact, the preterm birth rate in the US is really appalling. It's similar to some third world countries. So at the bottom, and I have to move my thing here. At the bottom, you see some colors. So the green represents less than 10 preterm births per 100 live births, and then the yellow is 10 to 15. So you can see that the US has the yellow color, as does countries like Sudan, Ethiopia, countries that we typically think of as having very poor healthcare systems. And the US shows a similar rate to these countries. And then other countries that we also consider to have poor healthcare systems and are under-resourced, countries like Venezuela and Colombia actually have a lower preterm birth rate than the US. So this is really appalling, and it hasn't changed much over the years. Even though the care for premature infants has improved, the premature birth rate has not. So this is a real problem. There are many factors that can go into what influences preterm birth, and so many that we can't get into them all today, but I'm just going to present one as it relates to my research. So first, let me tell you about the cervix. So on the far left here is a pregnant uterus, and the cervix is at the bottom of the uterus. And the cervix is firm early in pregnancy, and as pregnancy progresses, this structure changes in geometry and it changes in composition. So it softens, becomes less stiff, and we call that remodeling. And so that remodeling takes place so that at the time of birth, the fetus can pass through the cervix and be born. So accompanying those changes are changes in the composition and the structure, the microstructure of the cervix. So the cervix consists primarily of collagen, and these little circles you see here are fibers, collagen fibers, that are coming out of the screen. And you can see that early in pregnancy, they're very dense and well organized, and as pregnancy progresses, they become less dense and less well organized. And so this is the kind of evolution over pregnancy of the microstructure of the cervix. And so what is believed is that if this remodeling, this change in the stiffness and structure of the cervix happens too early, that can contribute to preterm birth. And actually, Professor McFarland, who is working with us on this project, does work with Professor O'Brien in ECE, and they look at quantitative ultrasound and how that relates to the structure of the cervix and the stiffness of the cervix. And in the clinic, they're showing that there's a strong correlation between this early change and preterm birth. I just want to go back to this image that I had on the first slide. Again, I said this is rat cervix. And so it's a cross section. So if you look at this orange line and you slice the cervix and then look at it, you would see a cross-sectional image. But I do want to mention that human cervices have one cervical canal and rat cervices have two cervical canals. And so this cervix that I'm going to be talking about does not look like a human cervix. And so I just wanted to mention that before we go on. Okay, so the goal in the work that I'm presenting to you today is to link the microstructure of the cervix, the collagen microstructure, and its mechanical properties, its local mechanical properties. And we're doing this in order to try to better understand how these relate to preterm birth and how we might use this information to prevent preterm birth. And I have in parentheses there and other collagenous tissues and other diseases, because these tools can of course be applied to other scenarios besides the rat cervix. So the tools that we're using, one is called second harmonic generation imaging, and we use that to quantify the microstructure. And then nanoindentation is what we are using for the mechanical measurements that we're doing. It's one method of mechanical measurements. And to do this, we're using another tool. We're using an animal model, and in this case, we're using a rat model. So SHG is a pretty established nonlinear optical imaging modality. It's unique in that because of the way that it interacts with collagen, it allows you to image collagen or get a signal from the collagen without any stain. So I'm showing two images here, cartilage and subchondral bone, both which have collagen. And the images you see here are optical images, but this tissue has been stained with a special stain that highlights certain constituents. And on the right, you see the SHG images that don't have any stain, and so you can just image fresh tissue. SHG has good spatial resolution. You can, depending on the tissue, you will have minimal specimen damage. And there are some emerging clinical applications that make it promising for use in the clinic as well. And then below here, I'm showing the fibrous structure of a normal ovary and one that has ovarian cancer, just as another example. I think I missed, no, I didn't, okay. And so we can use Fourier-Transform analysis to quantify the collagen fiber organization. And so I'm going to explain this in the simplest way possible, in part because I'm not an expert on this part, Professor Toussaint is, but I will give you the Amy version of this. So we take an image like you would see here, and we perform this Fourier-Transform analysis, which takes the data and puts it into what's called a frequency domain. So we can take a very simple image like this one here, and this is representing parallel fibers that are all the same width, they're all equally spaced. And if we do the Fourier-Transform on that, we get a line that's shown here. Okay, so that line you can see has a particular orientation, and that orientation of the line is related to the orientation of the fibers. In fact, it's rotated by 90 degrees. Okay, so we can do a similar thing with real images like you see here, where we see there's some fibers that are aligned. So we would divide this image up into little parts, like you see here, little subunits of the image, and perform the Fourier-Transform on each of those subunits of the image. And when we do that, we can obtain an orientation of the fibers in that little square, and then we can quantify that. Okay, so that's what lets us quantify the orientation on a very local level of these images. And if we do that, if we end up with, so in this case, we have this very distinct, sharp line that shows up, and that's because all our fibers are oriented exactly the same, and they're all exactly the same size. In this case, we have some variation in the image, and that would result in having something that looks more like an ellipse. And that ellipse would tell us that there's some dominant orientation, but there's still a range of orientations within that image. If this ellipse was instead a circle, then that would mean that the fibers are oriented equally in all directions. We would call that isotropic. If we get an ellipse, we call that anisotropic. So we can take the shape of this ellipse and we can use that to quantify the orientation of the fibers in our image. So SHG can also be done in 3 dimensions by taking image stacks. And so that's similar to confocal microscopy, where you can generate a 3-dimensional image. So we can do that, and we can do the Fourier-Transform analysis on all the images and end up with a volume where we can describe the orientation of the fibers. So these are images of tendon. which is a long tissue, right, like your Achilles tendon. And if we slice it along the length of the tissue, we would get an image that looks like this. And if we slice it at an angle, we might get an image that looks like this. So we can take those images, do the Fourier-Transform analysis on it, and these arrows then tell us how the fibers are oriented through that volume. So in the case of this longitudinal section, we see that the fibers are, the arrows are generally lined up in the same direction. There are a few that are a little bit off, but mostly in the same direction. Whereas in the case where we cut at an angle, we can see that the fibers in the volume are oriented at some angle that's different than the other orientation. Okay, so that's a little intro to SHG. And let me go back here. I'm going to change to talking about measuring mechanical properties, and I'm going to talk about nanoindentation, the tool we're using, in just a minute. But first, I want to explain why measuring mechanical properties of tissues is challenging. And I'm going to start by talking about bone, because I think bone is something that we cannot kind of all relate to more than some other tissues. So you can imagine your femur bone, for example, which might look like this. And you can measure the properties of that whole bone and get some value out. If you zoom in, I hope you can see my arrow. If you zoom in here, then you see some other structure. Okay, so I'm not going to go into the details of what the structure is, but you notice that there's some structure when you zoom in. If you zoom in even further, you see another structure. Okay, and if you zoom in further, you see yet another structure, and so on. And so we call that a hierarchical structure. And it's challenging because we don't know at what level we should be testing this material to get the properties that we're interested in. So that makes it challenging. The second thing I want you to notice is that we can see that if we're looking at the material here versus if we're looking here, that material looks different. So we call that heterogeneous. So it's heterogeneous through the bone. And then you can also notice that there's some directionality with this tissue, both with the bone that's shown here and the tendon shown here. You can see there's directionality to the tissue. So we call that anisotropic. And so if we test it in different directions, we might get a different answer. So in addition to those challenges, tissue is also wet and slippery, and it changes as soon as you take it out of the body. And so that presents additional challenges. Sample preparation can damage the tissue. And also it's hard to know when you're doing this mechanical test, what the structure is in the exact location you're testing. And then also, when you're doing tests, you're usually averaging over some volume. And that means that you're losing some information and some structural detail that could be important to understanding the properties. So these are all challenges that we face when we're testing tissue. So one technique to test the local properties of the tissue is called nanoindentation. And so nanoindentation, depending on the size of the probe, allows you to sample a small area of the material and measure the properties in that localized area. So some probes are several millimeters and some are several microns. The one we use is about 100 micron diameter. And so the idea with nanoindentation or any indentation is that you have a probe and you push it into the sample and you measure the force that it takes to push it into the sample. And then you also measure how far into the sample you push the probe. Okay, so that's the displacement D. And so you get out this force displacement curve. And what you're doing is, this is a simulation, you're pushing this probe into the sample and there's some local deformation that's occurring underneath the probe. And so we want to understand the relationship between this local deformation and the load that we're using to push the sample into the material. And so there are some models that we can use to back out some interesting information. This model is a very simple model. It's called the Hertz model. And generally it's used for very simple materials and actually generally used for more simple materials than I am showing you today. However, the models don't exist with this technique yet for these complex materials. So that is research that needs to be done. So we use this simple model, and we're measuring what's called the indentation modulus, this E star, and it depends on the force, displacement, and the radius of the indenter. So for those of you who know mechanics of materials, this is not the Young's modulus, this is the indentation modulus. And so I just want to make that clear that it's not the Young's modulus. So this is also typically used for small deformations and then simple probe geometries like a sphere. Okay, so we want to be able to coordinate or co-register our indentation data and our detailed microstructural data. And so we had to, we have two different systems. We can't measure these at the same time. So we had to co-register these data. And so here's the process that we used. This is a little cartoon of the rat cervix and the uterine horns. We cut the cervix, and if we look at it in cross-section, this is what it looks like in cartoon form. These were 12-week non-pregnant rats. We embed this in OCT, which is a compound that allows you to freeze the tissue, and it supports the tissue so that you can cut the sample without damaging it much. And we added some fiducial markers in here so that when we took images in bright field with the nanoindenter and took images in bright field in the SHG microscope, we could stitch those sets of images together and then co-register them and find features that we could align in both of the images. Okay, so that's what we did here, overlap bright field images. And then that allows us to correlate the microstructural information and the mechanical information. And we think we're within about 50 microns, 25 to 50 microns. So it's not perfect, but it's pretty good compared to anything in the literature thus far. Okay, so that's the process we use. So when we put those data together, our indentation data and our SHG data, we can construct what we are calling an optomechanogram. So this is the SHG image that I showed you before. And we have this blue line is where we indent across the sample every 25 microns. And then we use the data from indentation to back out the modulus. And so you can see there's variability in the modulus, but you see that in this region here, where it's less bright, the modulus seems to be higher. And in this region here, and in the brighter regions, the modulus seems to be lower. So we're going to talk about that in more detail as we go on. Okay, so the first thing we did is we took this 2D image that you see here. So this is 2D SHG. And using the Fourier-Transform analysis, we classified certain regions, and we selected certain regions to look at in 3D. Okay, and so we classified these as what we call near-septum region, that is somewhat isotropic, and the septum and ring regions, which are anisotropic. Okay. And here are 3D, one single image from the 3D stack of each of these. And you can see that the SHG images of the ring and septum are quite similar, and the near septum looks different. We can do this representation of the data like I showed you before, where we look at different volumes of the tissue, and we can show the dominant direction of the fibers in those tissue. You can see for the ring and septum, they're quite aligned. And for the near septum, there's a lot of out of plane orientations. We can also classify these as anisotropic and dark, meaning there's not much signal or there's not much collagen in that region. And we can also look at quantify the orientation of the fibers. So here I'm representing these as theta, which is the in-plane orientation, and phi, which is the out-of-plane orientation. So you can see that the ring and septum have similar in-plane orientations, and similar out of plane orientations, whereas the near septum is quite different in both those regards. So I'm going to look at those a little bit more closely next, in a couple slides actually. Okay, so here's quantified, the data quantified. So here I'm showing the dark percent, so where there's less signal, and the ring and the septum regions. And you can see again that quantitatively these two are very similar, but they're different from the near septum region. And we can represent that data so that you get a better sense of what that looks like compared to these values. If we look at the distribution of data and take two extremes as examples. So in this extreme, we see it's almost all purple, meaning that it's almost all dark. There are a couple volumes that have some anisotropic regions. And in at this extreme, we see there's a lot less dark, meaning there's a lot of signal from the collagen. And you can see the green, that it's mostly anisotropic at the other extreme. Oops. Okay. We can similarly look at phi, and again, quantitatively, the ring and the septum are similar, but both are different from the near septum. And again, we can take these quantitative numbers here and give you more of a physical representation so you can visualize what's going on at the extremes. So at one extreme, we see that the arrows are mostly green, which means there's a lot of regions that are out of plane. And the density of these arrows reflects the fact that there's not a lot of signal from the collagen. So the collagen density is quite low. And in contrast with this other extreme, we see that the arrows are very dense, meaning there's a lot of collagen signal, and that they're mostly red, meaning that they're mostly in plane, or their phi is less than 15 degrees. And you see a few green arrows here and there showing more out of plane data. Okay, we can look at two other parameters. One is called circular variance, and that's a measure of the dispersion or the variability in plane, the variability of theta. So we can quantify those values, and again, we see the ring and the septum are the same or similar, and they're dissimilar from the near septum. And we can again give you a physical representation of what those values mean at the extremes. So if you look at one extreme, you can see that these arrows are going lots of different directions, meaning that there's a lot of variability in plane. And in this other extreme, you can see they're almost all exactly aligned, showing very little variability in plane. And we can do, excuse me, just a second. We can do something similar, with something called spherical variance, that represents the dispersion out of plane, or the variability in both phi and theta. Again, we see the ring and septum are similar, and they're different from the near septum. At the extremes, we see arrows going every which way, showing there's a lot of variability, and here they're more aligned, showing less variability. So now the indentation data. Again, we see the ring and the septum show very similar indentation data that's different from the near septum. What is really interesting and was very surprising about the data that I'm showing you here is that the near septum, which had less collagen density, had a steeper slope. And so that's reflected when we take the load indentation data, or the load displacement data, and convert it to our indentation modulus. Again, the ring and septum are similar, and the near septum is different, but it has a higher modulus, meaning it's stiffer than the other two regions, which we did not expect. And we did this over 5 cervices, and I'm just showing the modulus data here, but all of the data that we collected for all of these 5 cervices were consistent. And so we do think that this is right, but I have to say that we don't quite understand the results as of yet. We looked at correlations, excuse me. We looked at correlations between the microstructural parameters that we got from SHG and the mechanical parameter. And we saw a positive correlation with each of these microstructural parameters that we measured with the modulus. And so, for example, with the amount of dark area, so less collagen, as the amount of dark area increased, the modulus increased, okay? And again, that wasn't expected. We expected that the more dense the collagen, the higher would be the Young's modulus. And so these other parameters similarly increased, and we do not have an explanation for those correlations yet. So I didn't mention at the beginning that this work is not published, and so I'm sticking my neck out here by presenting this work that we don't fully understand yet. So what is next? So we want to understand why these really dense fibrous regions appear to be less stiff by this measure of mechanical behavior. We also want to develop fibrous material models using this quantitative SHG data so that we can represent the mechanical behavior mathematically. Okay, there are a lot of fiber models in the literature, but none that are based on quantitative SHG data, and also none that would capture this unusual behavior that we see where we see a higher indentation modulus in these other regions. We also want to develop a method to incorporate anisotropy in this indentation approach and heterogeneity, because right now the models do not incorporate those two factors. The models are very simple. And then we also want to measure some other properties like poroelasticity and viscoelasticity. And then of course, the cervix that we have done this work on right now is non-pregnant cervix. And so we want to look at how these data evolve through pregnancy and whether we can really correlate those microstructural parameters with the changes in the cervix, and then hopefully be able to relate those to the quantitative ultrasound data and to data that's gathered in the clinic to better understand how this relates to preterm birth. So it's 35 after, and I was going to talk about another project we're doing on inflating small fetal membranes, but I don't really have time. I'll just say that we are doing that work on rodent models, and we have a miniaturized inflation device where this opening is about 1 centimeter in diameter, and we're using a profilometer to get a full field displacement measurement, and we validated this with other materials that we know the properties of. So with that, if you want to talk about that further, I'd be happy to talk about that offline. You can shoot me an email. And with that, I'd like to thank collaborators who've been working on this, and my student, Amir, who provided some slides for me. And I'd also like to thank the funding agencies and of course the IGB and MRL where this work was done. So with that, I'm happy to answer any questions that people have, and thank you for your attention.
Great. Thank you very much, Amy. We're now open for questions. If you'd like to type them in the chat box, I can read them. Or if you'd like to use the hand raising mechanism in chat, you can do that and then speak your question. I have one that came to me. I'll start off, Amy, and
Sure.
that is, how long does it take to modify some of these stiffness parameters, like with nutrition or stress or things like that? How quickly do they change?
That is an excellent question that I do not have an answer to. So we have not thought about trying to do this for the cervix in relation to pregnancy. But one, I think we will be probing this when we look at environmental toxins and the effect on reproductive tissue. So there, I think we'll be looking at ovaries and how that affects fertility. I'm not sure that we'll be looking at that for the cervix initially, but I don't know how long it takes for turnover of these tissues for that to play a role.
Mm-hmm. A question came in, another question. Are you sectioning the tissue? And if you are, how thin are the sections?
So we have to keep the sections fairly thick in order to do the indentation. So if you were sectioning for histology or regular SHG imaging, you might do something like 50 microns or 10 microns or something like that. For us, because we are using the sample for indentation, we have to have it be 50 to 100 microns thick, sometimes more, because if it's too thin and you start to indent, you start to interact with the hard substrate below, and then that affects your result.
Next question. This one asks, are you concerned about percolation of the embedding epoxy into the near-septum area, leading to an increase in effective stiffness?
So we don't use epoxy, we use OCT. And that is a material that freezes at relatively low temperature so that we can cut it, but then you remove it from the sample entirely before you do the imaging and the indentation. So to our knowledge, that does not affect the tissue, and it's a very common technique for people to use to cryosection tissue, but it's not epoxy. Epoxy for sure would affect the stiffness of the tissue, no question.
Next question, how was the tissue constrained during the indentation tests?
Good question. So, so we make it, and this was a big problem actually, because the tissue would just float away and make it really hard to test. So we put a little bit of super glue, a very thin layer of super glue down. And we have tested to make sure that when we indent, we're not interacting with that harder layer underneath. So we make it thick enough to avoid that.
Next question, is it possible that nanoindentation performs worse on fibrous materials, thus explaining the observed difference in stiffness?
So I'm not sure what worse means for mechanical testing, but I think one of the contributors to the difference in stiffness is that we can only see the collagen. And there's another important constituent there called glycosaminoglycans. And glycosaminoglycans are known to be a constituent that behaves differently in compression and tension, and similarly fibers behave differently in compression and tension. So gags are an important component of, say, cartilage, which has really good behavior in compression. And so these molecules resist compressive loading for a number of reasons. But what we think is happening, and we haven't been able to verify this yet, but we think that the GAG content is higher in the regions that are showing higher modulus. And because this indentation method has a lot of compression in it, we think that that is why we're seeing the higher modulus. Again, that's a hypothesis. We haven't validated that, but that's what we think is happening. It could also be that, you know, we're using this model that is intended for much simpler materials, and we're using them on a really complex material. And so it's possible that if we had a model that could incorporate anisotropy, then we might have a different outcome there. And we'd have to model that, and we haven't done that yet.
Next question, is it possible to use the RAP model and a combination of imaging techniques and genomics to find blood markers of stiffness?
Interesting. I have no idea. That's an interesting question. I absolutely have no idea. Mm-hmm. Good question.
Okay, any other questions? Here's one. Have you tried AFM to see the collagen versus GAG fibers, or environmental SEM to try to image them? And maybe you can unpack the acronyms for the audience.
Sure. So AFM is atomic force microscopy, and it's another way that you can probe the surface of the sample, but it offers both mechanical information and also some imaging information. And people have used AFM to image collagen. I have that image somewhere. Dr. McFarland, I don't know if she's still here, but she has some of those images. You can see the collagen. You can't see GAGs. I think you would have to use, the GAGs are the glycosaminoglycans, by the way. Those are the molecules that work well in compression, that resist compressive forces pretty well. So I don't think that you can image those without using a stain. And so AFM would not allow you to image the GAG molecules, to my knowledge. You would have to use something that would stain so that you could see in fluoresce or other optical modality, or you could use electron microscopy and use a stain, but I don't think you could see them with AFM.
Okay, any other questions? All right, well, I'm sure everyone means to join me in thanking you for a great talk. And thank you everyone for coming. Thank you, Amy, for speaking, and please be safe and be healthy. Bye-bye.
Right, thank you very much for the opportunity. Take care, everyone.
Bye-bye.
Bye-bye.